In this video I'm going to present a COSBI contribution to Venus C. In fact, I'm going to show you how the Venus C framework has been applied to the field of systems biology. On one hand, in this scenario, we wanted to port on Venus C the tools that were previously developed by COSBI. On the other hand, we plan to provide the researcher with a tool that could leverage the power of the cloud. Systems biology studies the dynamics of complex systems made of several components interacting with each other. The representation of a system is called model, and the parameter of a model are the initial concentration of the components and the frequency with which they interact. Models are simulated in order to see how concentration of components vary in time. We asked our researcher which are the most common experiments they have to deal with. We've come up with two measure classes. To the first one belong sensitivity analysis experiments. The goal of such experiment is to understand how perturbing the model parameters can affect the response of the system. The second class is made of model fitting experiments. Here the researcher knows the logic of the model, but some or all of its parameters are unknown. The goal of the experiment is to find the configuration of the parameters that most closely matches the wet lab results provided by biologists. While on the sensitivity analysis experiments there was a common agreement on the data flow to be used, the same was not true for model fitting experiments. Here the data flow can change because of researcher preferences or because of characteristics of the problem tackled. We decided to provide the researcher with a straightforward way to run sensitivity analysis experiments, but instead of forcing our decision on researchers, we allowed them to customize their model fitting experiments. We have chosen a research project for each of their scenarios as use cases. For the sensitivity analysis, our intermediate scenario, we have chosen a model of gemcitabine, a drug for cancer treatment. Here, the perturbator parameters could have been the frequency or the amount of the drug intake, or the expression of some protein. For the model fitting, the advanced scenario, we consider the model relative to one of the most studied transcription factors, p53. COSBI researchers collaborate on a daily basis with several research institutes, both public and private, in the fields of biology, pharmaceutics, and medicine. At the moment, the two projects we already mentioned are using our platform, but 5 more are planning to use it. In order to support the two scenarios, we provide 2 different architectures. For the intermediate scenario, the COSBI framework may rely either on the generic worker or the compass independently of the cloud provider. In this case, the only input needed is the model on which the experiment should be run and the initial parameters. For the advanced scenario, the use of the generic worker on Azure is required, and the framework provides a further layer that allows the researcher to submit, along with the model, an Iron Python script describing the experiment data flow. We then decided to figure out the amount of resources we could need. We found data that both for gemcitabine that required 64 simulations and p53 with more than 5,000 simulations, we could obtain a speed up of 10 with 25 cores. We considered these results satisfying, in particular for having reduced the running time of the p53 experiments from 29 hours to 2 hours and a half. Let's see now how the data flow of the model fitting experiment looks like and how the experiment is executed on the platform. The model of the p53 is given an input by the researcher, along with a script and some custom information needed by the script. When the experiment is submitted to our platform, only a special job is created, here called Sparkle and represented by a box with a red border. The Sparkle job interprets the script to create the real data flow of the experiment by submitting new jobs. The first job that is submitted creates several parameter configurations for the model, again using the script. Then, for each configuration, several simulation jobs are submitted. The last job is in charge of collecting all the results from the simulations and, according to the script, computing a measure of goodness for each configuration. The produced output is a small CSV file that will not affect the network transfer accounting. To wrap it up, from COSBI point of view, we provide researchers with a platform that is simple to use for the most frequent experiments and yet allows the creation of ad hoc system biology experiments. Such flexibility was possible thanks to the integration of power scripting environment like R and Python. The use of the cloud allowed the reduction of the running time of the experiment, allowing larger experiments to be run, leading to a finer accuracy of the results. Furthermore, we consider an added value the possibility of scaling the resources according to the real demand. On VNC's side, we appreciated the fact that a basic porting of our tools was straightforward, and also the creation of clients for submitting our jobs was an easy task. The concept of data flow allowed us to abstract from the storage. In fact, the input-output parameters are transparently mapped to the files to be transferred locally to the machine that is running the job. In particular, generic worker allowed the dynamic submission of the jobs, and this was fundamental for the creation of our dynamic data flows. Finally, we could easily create a very simple job descriptor to be used by the scripts by simply wrapping up the descriptor of the generic worker. Thanks for your attention. I want also to remind you that the videos of the demo of our platform are available.